Hello, everyone. Thank you so much for joining us today for another e-seminar in Translational Biomedical Engineering. We are very happy to host Professor Kathryn Grandfield from McMaster University, who's talking about multi-scale characterization strategies to probe osteointegration and biomineralization. So this talk is also sponsored by the Canadian BiomaterialsSociety. So next week, we will have our another colleague from McMaster University, Professor B.Eng from the Department of Chemical Engineering and the School of Biomedical Engineering, who is talking about platform technology for high throughput and high content drug screening. So for getting the most updated information about the e-seminar series that we started in June 2020. So you can always follow our Twitter account with the hashtag TransLBME. And then, last but not least, we want to thank our sponsor, Montreal Transmedtech Institute, who sponsors this talk, this seminar series, and also Canadian BiomaterialsSociety for this for today's talk. So by that, I introduce our speaker. Professor Grandfield is an associate professor in the Department of Materials Science and Engineering and the School of Biomedical Engineering at McMaster University. She holds the Canada Research Chair in Microscopy of Biomaterials and Biointerfaces and is the president of the Microscopical Society of Canada. Professor Grandfield received her Bachelor of Engineering and Master of Science in Material Science and Engineering from McMaster University and her PhD in Engineering Sciences from Uppsala University in Sweden. She then completed the postdoctoral fellowship in the Department of Preventative and Restorative Dental Sciences at the University of California, San Francisco. She is the recipient of several awards, including the 2017 PetroCanada Young Innovator Award, the 2018 Early Researcher Award from the Ontario Ministry of Research, Science and Innovation, and 2019 the McMaster Faculty of Engineering Teaching Excellence Award. She served as the inaugural Director of User Operations for the Canadian Center for Electron Microscopy, as well as the past secretary and member of the board of directors for the Canadian BiomaterialsSociety. Her current research interests center on biomineralization and span the development of new biomaterials in additive manufacturing and the advancement of characterization strategies using multidimensional electron and ion microscopy. By that, I invite her to share her screen and start her presentation. Thank you so much.
Great. Thank you very much for the introduction. Can you see my screen now?
Yes, that's perfect.
Okay, excellent. Great. Well, thank you once again for the introduction and the invitation to speak here today. I'm really excited and honored to share with you the work that we're doing at my lab at McMaster University. And to begin, I'll tell you a little bit about the type of work we're doing and where we're located. So we're in Hamilton, Ontario. And at McMaster, we're fortunate to have the Canadian Centre for Electron Microscopy and the Brockhaus Institute for Materials Research. And so we have access to quite a number of advanced electron microscopes and infrastructure that's really world class. And so my research program has taken advantage of this opportunity and uses a lot of electron and atom probe microscopy in order to study biomaterials. And the types of biomaterials we're really interested in studying in my group are all bone interfacing biomaterials. So we study things like dental implants, orthopedic implants, and we're also interested in studying biomineralization in natural tissues and the development of new biomaterials, such as materials made from additive manufactured metals. So today I'll be talking to you a little bit about how we're developing multi-scale microscopy approaches to look at biomaterials interfaces and to look at some biomineralization research that we've done in our lab. And because there's a number of trainees in the audience, in the introduction, a little bit of my path from where I started to where I am today has already been shown, but I wanted to highlight for the trainees a little bit about my past and my path through research and towards academia. And so it really began when I was an undergraduate student at McMaster. I was fortunate to do research internship during 2 of the summers that I worked there. And that led me on my passion for research and eventually to do my PhD in Sweden at Uppsala University. And for those trainees in the audience who have the opportunity to travel abroad during your studies on even a short exchange visit, I highly recommend it. I think you learn a lot by traveling and researching in new locations. Just after I finished my PhD, I was a visiting researcher at the University of Antwerp for a short period of time before I did my postdoc in San Francisco, and then returned to Canada and started my research group up in the Department of Material Science in the School of Biomedical Engineering. So I'm really excited that I'm at McMaster doing the work that we're doing in our lab today, and to share that with you. So why are we interested in studying bones? So there's many things we could study in the body. For me, I find this quite fascinating because of the number of people that it affects. So if we take a look at some bone diseases that are quite common, one of them is osteoporosis. This affects 20% of women over the age of 50. Other diseases that might impact our bones would include having a huge number of spinal fractures as a result of osteoporosis. So if we look at stats from the US, 1.5 million new spinal fractures happen annually. We also have a huge number of dental implants placed every year, about 1 million in North America, but with only about a 95% success rate. And that seems quite high, but if you're in that 5%, 5% of a million is a large population that's having an implant placed that's failing. And if we look at projections for the future, we see things such as total joint replacements, hip and knee replacements are on the rise. So hip replacements have grown by 35% in 2020, and knee replacements are expected to grow by 400% by 2040. This isn't to mention all of the other possible things that we could have affect our bones. So cancer, rare bone diseases, hyperglycemia, diabetes. So there's a huge need to not only understand bone structure, but to understand how it interacts with biomaterials and therapeutics so that we can prevent some of these diseases, and in many cases when we can't prevent them, to treat them. So, bone's also a fascinating material just from a material science perspective. So my background is in materials engineering, so I find it to be fascinating as a material itself. And when you zoom in at bone, you can see it has this really unique hierarchical structure that spans all the way from the macro scale to the atomic scale. So on the far right, we see the macro scale structure of bone, and we have a cortical shell that's quite thick and dense, and then we have a spongy trabecular interior. And when you zoom into bone, for example, in the cortical bone, you'll see these round features called osteons, and these align themselves along the length of a bone. And embedded within the osteons is a complex cell network of osteocytes, and these are responsible for signaling other cells to deposit bone and remove bone so that it's always remodeling throughout our life. And as you zoom down to smaller length scales towards the nanoscale and even atomic scale, we have hydroxyapatite crystals and collagen fibrils making up our bone structure. So we can see that we have this continuum of structures that spans several length scales. And in order to understand bone itself and treatments for bone diseases, we need methods that enable us to visualize across the spectrum of length scales. And that's what we're really trying to achieve in my lab. And we're trying to achieve that with some of these tools that you'll see on this slide here. So again, going from the macro scale all the way to the atomic scale, there's three-dimensional imaging tools that we can use to visualize bone. So you'll see some of these acronyms today in my talk. So I wanted to give you a brief overview of them. You'll see things about X-ray computed tomography, focused ion beam scanning electron microscopy, scanning transmission electron microscopy, and atom probe tomography. And specifically, I want to show you 3 examples of how we're using these tools to develop imaging strategies for biomineralization. In the first example, I'll show you how we're looking at bone implant interfaces and asking the question about what is the nanostructure of the bone implant interface. In the second example I'd like to cover, we're looking more at the nano and atomic level and trying to understand the collagen mineral arrangement in bone. And the last example I'd like to share with you is looking at bone at the mesoscale and really trying to understand the structure of bone across microscale volumes with really nanoscale resolution. So let's get started. Let's dive into the first topic, taking a look at bone implant interfaces and really trying to understand the nanostructure of the bone implant interface. And this is some work that we've done in collaboration with colleagues in Sweden and at the Canadian Light Source and in France as well. And the main work of one of my previous PhD students, Xavier. So to give you a bit of an introduction, why do we care about osseointegration, or what is osseointegration? Anytime we have an implant placed in the bone, we're concerned about the contact and the bond that osseointegration or that the implant makes with bone. And this is what we call osseointegration. Historically, it's been defined as the direct contact or bonding between a bone and an implant. And this definition was really on a light microscopic scale, so at low resolution. And we're interested in looking closely at what happens when an implant is placed. So an example here, we have a piece of bone with an implant site drilled into it and an implant placed in the center. And one of the first steps we have would be recruitment of cells to the implant surface, deposition of the initial stages of bone growth by these cells, and these cells continue to grow bone away from the implant surface in something called contact osteogenesis. If we zoom in on these bone implant interfaces, we can see some interesting features, cells embedded at the interface. But our question is really, what is this stuff right at the interface between the bone and the implant? What's attaching the two together? What does this look like? How does its structure change when the implant itself changes? And we really want to probe on what level does osseointegration occur. So is it just a microscale phenomena of contact between bone and implant, or is there something on a nanoscale level that's taking place? Now, osseointegration is very complex. This cartoon shows a very simplified view of it, but there's many processes taking place at once inside the body. And there's many things that I won't have a chance to talk about today, and some of them are shown here. That's the important role of proteins at the bone implant interface. We know proteins are mediating the cell response to the implant. The role of vasculature is also really critical in how much bone and what types of bone will be forming at the implant interface. And of course, cell networks, so how our cells are reaching out and touching the implant interface. So these are all critical to consider, but today what I'll talk to you a little bit about is what we see with the bone structure and chemistry, so more from a structural perspective. So how would one assess osseointegration clinically or conventionally? Well, if you went to your dentist after you had a dental implant placed, you could have a radiograph, and this would show the contact between the implant and the bone and any major problems with the bone tissue itself. In order to study this further, we need to have the implant out of the body and look at things closer up. So using light microscopy, histology is one of the main ways in which we evaluate implant success. And in this case, we have an implant in black and we have this bone tissue in purple, and we can measure things such as implant contact and implant area. We can move to higher resolution techniques such as scanning electron microscopy, and again, see the thread valleys of a dental implant and new bone tissue growing in and where it's not attaching to the implant as well, we can see. And lastly, to look at bone on the nanometer scale, we would take this implant and put it into the transmission electron microscope, where now we can see features such as individual hydroxyapatite crystals and collagen banding next to the titanium implant. But the challenge with this is that these are all two-dimensional imaging techniques for what's an inherently complex and three-dimensional structure. So we really want to look at 3D microscopy techniques to probe bone implant interfaces. And again, on the same length scale, there's a few ways we can do that. So instead of just taking a radiograph, we can do X-ray computed tomography. So here's an example where we can see an implant and a 3D structure of bone around it. We can also look using focused ion beam microscopy to look at implant bone interactions. And at the nanoscale, we can use electron tomography to see how bone is attaching to an implant. And so these are some of the tools we've used in my lab, but they're still lacking something. These are 3-dimensional techniques. So we have X, Y, Z information, but what we really wanted to add to this in my lab was chemical information and approaching a 4th dimension where we have our 3D spatial space and our fourth dimension is chemical information. So what I want to show you today is how we're using 2 types of strategies: electron energy loss spectroscopy tomography. That's a huge mouthful, so you'll hear me say EELS. And we're also using atom probe tomography to look at how bone and implant are acting with respect to one another. So the example I'll show you is as this biomaterial here. This is a titanium implant that's been laser modified. It's used in dental applications, but also in bone-anchored prosthetic devices, as you'll see on the left here. And the thread values of this implant have been modified to have a roughened nanostructure. And this is a titanium dioxide-rich structure that forms on the surface after laser ablation. And when these implants were removed in preclinical studies, we found that the force to remove this implant with modification was 150 times higher than to remove an implant that had no modification and was smooth. So we really wanted to understand what is it about this nanoscale structure that's improving osseointegration, or this contact between bone and implant, and causing these high values of removal torque. So we wanted to look closely at this nanostructured implant surface in bone. And in order to do that, we need to make a small sample. The transmission electron microscope will only allow us to have a sample about 100 nanometers thick. So we need to use another microscope first to create a specimen for the TEM. And here's where we use something called focused ion beam microscopy. It's like a scanning electron microscope, but instead of just having an electron beam for imaging, we have a gallium ion beam where we can insert ions and mill material away inside the microscope. So this is something what the workflow would look like. We'd identify an implant and bone touching it that we're interested in. We deposit a protective layer of metal inside the microscope and then use these ion beams to mill out, like a little sand blaster, the area that we're interested in. We can then use a small micro manipulator to pluck this section out. And then we use the ion beam again to thin this section until it's less than 100 nanometers and we can see through it with a transmission electron microscope. Then we take this to our TEM, and this is one of the TEMs at our facility shown here. And we use our electrons that now go through this thin sample and create an image. And so on the right-hand side here, you'll see this titanium implant, the nanostructured oxide layer that's on the surface of it, and bone structure that's above it. And what you'll see in these TEM images, you'll see this banding pattern. So this dark and light overlapping pattern, and this is a result of the collagen structure in human bone. So anytime you see a TEM image of bone, you might expect to see this banding pattern because of the organization of collagen. So this is interesting to see it in 2D, but I promised you 4D, so we'll get there. So let's take a step further and look at 3D first. So inside the TEM, we can take this thin section and we tilt it. So you can imagine it's something similar to X-ray tomography, except we're using electrons. And when we perform this, we can create a reconstruction by similar methods of algorithms to reconstruct 2D images into a 3D volume. And now what we achieve is a 3D structure of our bone implant interface that has nanometer resolution. So we can more clearly see these collagen fibrils and how they move across and interact with the implant zone at the interface here. Now, This is exciting to have 3D information, but there's a bit of a problem with this type of acquisition, which is the standard acquisition in TEM tomography. And that's something called the missing wedge. So you'll notice in this cartoon, there's an area we can't tilt to. There's not enough space in the microscope for us to tilt a perfect 180 degrees. So we result in this area that we can't access and probe, and this causes some stretching in the 3D reconstructions. So ideally, we'd like to rotate a perfect 360 degrees, and we can do that by performing something called on-axis electron tomography. And in this case, instead of having a thin, flat sample like I showed you, we make a thin cylindrical sample, which we can rotate inside of the microscope. So to do that, we go back to our focused ion beam, we identify an area we're interested in, and instead of taking our lift out and milling it as a rectangle, we begin to mill it into a cylinder and make quite a thin cylinder that we can put back into the transmission electron microscope, and now have a needle that we can rotate and perform some other chemistry on because it's a uniform thin needle. So we have this needle, we put it back in the microscope, and we can take images and create a 3D volume. So here's the 3D volume shown here, our titanium implant on the bottom. We can see this nanostructured oxide on top of it and bone tissue above that. And so as we tilt this sample completely through the electron microscope, not only do we acquire images, but now we can acquire elemental information as well. We start to acquire elemental maps for different elements. So the ones we've chosen in our work are carbon to represent collagen. We have calcium and phosphorus to represent our bone mineral phase, hydroxyapatite. We have titanium as well as oxygen we're collecting. So at each angle, instead of just taking an image, we take a spectrum map and we can plot out in 2D where all of the elements are contained. So we repeat this over all of the angles that we're able to tilt to, and we can record and reconstruct these elemental maps into a 3D volume. So now we have a volume that isn't just the images put together, but we have all of the elemental signals on top of one another. So we'll just see if this video will play. It's frozen a little bit, so I'll speed through it for you. So you can see in this image, we have now the possibility to turn on where our elements are. So in red, we have where our collagen fibrils are, and we can turn that signal off and turn on our calcium signal and understand where our calcium is located with respect to the interface. And so this is really exciting because now we don't just have a three-dimensional image, but we have a three-dimensional chemical structure, or what we could call a four-dimensional data set. So with a four-dimensional data set, we can explore it. So for example, we can take this volume and we can take a slice anywhere within inside the center of it, and we can look at the chemical signals. So we can see our calcium containing zone. We can see a fibrillar zone where our carbon is representing our collagen fibrils. And we can look at where our titanium dioxide is located. And what's quite interesting is we can note that there's this afibrillar zone just above the titanium dioxide and below the collagen, where we don't have collagen fibrils, but we still have a calcium signal. So we've noted this afibrillar zone at the interface. One thing that's quite hard to tell, though, is what's happening in here where the titanium dioxide is. We really wanted to know, does the bone-like mineral just stop outside of the oxide layer, or is there an integration of bone all the way into the implant and into the fine features of the nano-oxide? So we couldn't probe this with eels. It wasn't high enough resolution, although it's quite high. So what we wanted to do was take this same sample and probe it with another technique called atom probe tomography. So an atom probe microscope is a really interesting microscope. You take a cylindrical shaped sample that has a really pointed tip, so similar to what we've already used. And by applying a laser pulse to the end of the sample, you can create field evaporation. And in this case, you'll have ions or atoms ionize, and they move towards a position sensitive detector. and that's also a time of flight detector. And we measure the mass to charge ratio of each of these atoms and the time it took for them to hit the detector. And we can create a three-dimensional map of where every atom was in the sample. So we've applied this technique to look at bone structure. So it's a mass spectroscopy technique. So when we record the raw data, you'll see something like this graph on the right with many of these peaks showing all of the different mass to charge ratios of each of the ions and molecules that evaporate off of the sample. And by doing some data analysis, we're able to separate out the signals representing bone and representing the organic phases of bone, such as collagen, and the mineral phases of bone, such as hydroxyapatite. And we can create a three-dimensional map showing the location of each of these species as well. So when we take our same sample that we did electron tomography on and EELS tomography, and we place it in the atom probe, we have a very small volume that we collect right at the bone implant interface. And each one of these dots here represents a cluster of atoms. We have titanium atoms at the bottom. We have our calcium and carbon atoms at the top here, which are representing our bone mineral. And then we have a layer showing our oxide from the laser ablation. And what was interesting is we noted that titanium dioxide was on top of actually a nitride surface underneath. And this was due to our sample being laser modified in air. But because we have this map of where all the atoms are, we can perform a lot of calculations with this. And so one of the things we can do, this is another sample showing titanium, our oxide layer, and then our calcium and our carbon just above it. We can perform a line scan across this 3D volume and map out the different chemical compositions. And so what we can see when we do this, we can see that there's a strong intermixing of our titanium from our titanium dioxide and our calcium phase across quite a broad interface. And so we see that we really have mixing of our calcium bone-like mineral into our titanium dioxide. We also see sodium and magnesium in high concentrations at the interface. And we really have this atomic continuum. So we don't have an abrupt interface where the bone is stopping outside the oxide layer. Rather, we have a penetration of the two layers together. We've looked at some other imaging techniques or spectroscopy techniques to see what type of mineral could be inside of the oxide layer, what type of mineral could be away. And some of these techniques show us that it's likely that we have at the interfacial zone more of an amorphous type of calcium phosphate that's introduced inside of this oxide layer. Whereas further away from the interface, we have the standard type of hydroxyapatite crystal. And this work shows something quite similar, that we have a different calcium species at the interface. So this is work done at the synchrotron in Saskatoon. And we also can note that there's some titanium leached into our bone just from this interface that we looked at as well. So this strategy is quite interesting. It shows us that we have a way that we can go from all the way from the 3D scale, trabecular bone structure, using micro-CT, we can see large-scale interactions of osseointegration. But by moving towards the nanoscale using electron tomography and EELS tomography, we start to approach information about the fibrillar structure of bone, so where the collagen fibrils are located and where we have an afibrillar zone. And then using some of our more advanced atom probe tomography and spectroscopy, we can begin to see that this intermixing of bone mineral and titanium dioxide takes place, and we really have an atomic continuum of osseointegration when we have an implant attaching to bone. So this is exciting, and some of the other work in our lab is looking at applying this workflow to different types of surfaces. So this was a laser modified surface, but we're investigating what happens when that surface contains drugs, or what happens when that surface contains other nanofeatures, such as nanotubes, for example. Okay, so let's continue on this idea of looking at the nanoscale And I think some of this work we did at the bone implant interface made us question, well, what is really the nanoscale arrangement of bone without an implant in place? And so that's where we wanted to look at the collagen mineral arrangement of bone. And this is some work we've done with some of our collaborators at McMaster University. And so there's several models that exist for bone structure on the nanoscale and for the collagen mineral arrangement in bone. And I like all of these models because they employ electron tomography. So it's quite interesting that all of them have used the same tool to understand the arrangement between collagen fibrils and hydroxyapatite crystals. One of the first models of bone by Landis et al. Shows that the mineral phase begins inside this gap zone between collagen fibrils and nucleates outwards. A model by my colleague at McMaster shows that instead the mineral takes the form of curved plates wrapping around the collagen fibrils. And just a few years ago, a model by Natalie Reznikov, who's now at McGill University, shows that the mineral phase takes place in this cross-fibular mineralization of these slightly different shaped particles. So all of these look at the structure, but they're missing one key thing, and that's the chemistry. So we thought, can we apply some of our tools, such as atom probe tomography, to contribute to understanding the collagen mineral arrangement, taking into account not just the 3D shape, but also the chemistry of what we're imaging. So we prepared samples of human bone, and we looked at this. This is an example of what it looks like inside the microscope, and we place this sample in the atom probe microscope, and we evaporate those atoms as I described previously and create a three-dimensional map. And we're able to have good correlation between our images and our 3D chemical data. And when we look at that over a larger scale, we see an example here from the atom probe microscope. And we can explore this data down any direction in any portion of the data set. So here's two slices I'm showing. And what we see are some features where we have calcium-rich plates, and we have carbon-rich gaps in between. And in other areas, we really see these looking like circular features with a collagen fibril in the center. And so this aligns quite well with what we see in transmission electron microscopy when we're looking at bone. In one orientation, we see this nice collagen banding in our bone structure, similar to what we see in B. And in another orientation, we see this curved morphology of the hydroxyapatite plates around collagen fibrils. And that's quite similar to what our atom probe data is showing us as well. We can explore this data a little bit further, and what we see is that we have co-localization of some trace elements such as sodium, which is an important element in some proteins, with the collagen fibrils. And we also found that at interfaces between collagen and mineral, we see a peak in sodium, also perhaps suggesting the role of proteins in templating the growth of hydroxyapatite along collagen. But we're missing the information about the orientation of collagen. The previous example I showed you was from the maxilla, and we didn't know the loading direction of the bone. So instead, if we could take a sample where we know the orientation of the collagen fibrils, we could have a bit of advanced information about our sample. And this is possible if we take a long bone. So in this case, by using a femur instead and slicing samples down perpendicular orientations, we know in advance the overall orientation of the collagen fibrils. So we did this to create an atom probe sample. We took a sample where we knew we could align the collagen fibrils along the length of the atom probe needle. This is an example of the image here. So we see our collagen fibrils and our hydroxyapatite fibrils are aligned roughly along the length of the atom probe needle. And then we perform atom probe tomography on this sample where we know ahead of time the orientation. So what we can see here is we can see this 3D data set where we have carbon, which is our collagen-rich phase shown in silver, and calcium, which is our hydroxyapatite mineral shown in teal. And we can explore this in 3D. And what we can do is we can decide to probe closer into one of these regions. So we can say, let's take a look at this collagen fibril here. And because we have the data in 3D, we can crop out a small set of the volume and explore it in more detail. So this is one of those small sets we can see here with great detail. We have a collagen fibril that we can remove and start to probe and explore where the calcium is in relation to the collagen itself. And what we see here is that we have evidence that looks like the calcium is surrounding and also integrating into some zones of the collagen fibril. But can we look at that in some different ways? We certainly can. One of the advantages of atom probe tomography is we have a number of ways to visualize the data. So here we show the data in heat maps for carbon, which would be our collagen fibrils. So those are bright red regions that appear to be collagen fibrils, and calcium, which is our mineral phase. And an analog to this in the lab would be in vitro models. So a lot of people are studying collagen fibrils that have been extracted and mineralized in a petri dish, and what they can see is quite nice information about how mineral interacts with the collagen. But the challenge with in vitro models is that all of these are mineralized on the lab bench. So the observations we make may not be exactly the same as the way that collagen is mineralized inside a living species. So by using atom probe tomography, we can actually pluck out one of the collagen fibrils and explore it in more detail. And now we have a collagen fibril that we know has been formed in an in vivo system versus in an in vitro system. And when we explore these with some other visualization techniques, here's a single collagen fibril that we can look at in more detail. We can see where our carbon is. We even see some of the subbanding inside of the collagen fibril. And if we look at where the calcium and phosphorus are located, representing the mineral, we see that there's a predominant amount of mineral exterior to the collagen fibril, but we also see places where the mineral integrates inside the fibril. And when we probe this and look at the spectra, we can extract spectrum from the atom probe for distinct regions. So we can say, what's the spectra outside here in this zone outside of a collagen fibril, and what's the spectra inside? And when we look and compare those, we see that the mineral inside of the collagen fibril appears to have a calcium phosphate ratio more similar to amorphous calcium phosphate, and exterior to the collagen fibril, we see a ratio more similar to hydroxyapatite. So this is giving us some indication that we have both phases of mineralization taking place. We have mineral inside the fibril and mineral exterior to the fibril, but perhaps these two types of mineral have a different phase. And so by using this, we now have an interactive 4D system when we create an atom probe data set that we can really look through and analyze the location of different elements and even trace elements in bone tissue. So we're excited to use this further. Some of the work in my lab is exploring the effect of drugs on bone structure and looking at some bone diseases to understand how mineral is changing with some disease. Okay, so the last section I wanted to talk to is to take a step back from the nanoscale and go to a much larger scale. And so this is really the mesoscale of bone. And what we're interested in looking at here was understanding how bone structure looks at higher length scales. And this is something we achieved using a new microscope at our facility called a plasma-focused ion beam microscope. And we were one of the first groups to apply this to human bone. So as I showed at the beginning of the talk, we have this structure of bone that's really unique and hierarchical, and there's been a lot of advances at imaging different parts of the bone. Some of them we've seen today. So if we're interested in looking more at smaller length scales, atomic scale, and nanoscale type resolution, scanning transmission electron microscopy tomography is a great tool for this. So here's an example of a STEM data set from our lab of bone structure. What you can achieve is high resolution. You can look at mineralized tissue or demineralized tissue. but it has a major drawback that it's a very small volume, less than 100 nanometers in thickness, maybe 200 nanometers. So we're missing a lot of the structure, and we're only looking at a very small region, a needle in a haystack, you could say. To look at a little bit larger networks, focused ion beam is one of the tools you can use. So instead of using it to prepare samples for the TEM, it can be used in a 3D imaging mode. This is an example of work done by Natalie Reznikov. And here you can see collagen banding really clearly. So you have a slightly larger volume, tens of microns. You have intermediate resolution. It's quite good, the resolution of an SEM. And this data set shown here is on demineralized bone, so it's missing the mineral component. But if we want to probe the mesoscale, this really means covering the microscale volume all the way to the nanoscale. And this would encompass things like full cell networks and other details that are hard to view when you're looking at such small regions as were shown previously. So to access this, we're using a plasma fib, which gives us quite good resolution, the largest volume that we can achieve with high resolution in an electron microscope, and allows us to look at different types of bone. So what is this plasma fib tomography technique or serial sectioning? Well, what we do is, like the focused ion beam I showed earlier to prepare TEM samples, we have an electron beam, but we also have an ion beam that in this case is xenon, which mills material much faster than gallium. And so what we do is we use this ion beam to mill away a slice of material. You can think of it like slicing a piece of bread. And we cut away a surface of the bread, and then we take an image. So we repeat this over and over. You can see the sequential milling and imaging. And then we stack up these images together and create a 3D reconstruction. And the advantage of using a plasma fib or a xenon-based fib is that we can access much larger volumes than using a smaller gallium ion beam. So this is an example from other work where you can see a comparison between the two volumes that you can access with these two tools. So for bone, that means something quite exciting. That means where a traditional fib SEM with gallium, you might see part of a cell and some cell processes. When you apply plasma fib SEM, you can start to see several cells and the interaction between the cell networks over a much larger volume. And so in our work, we've also applied this to looking at demineralized bone, like was done previously with regular fib SEM, and also mineralized bone, keeping all components of bone intact. So I'll start by showing you what we see when we look at demineralized bone. And in this data set, we're showing approximately 60 microns by 50 microns by 10 microns thick. And what you can start to see are some details about the bone structure. So in the top left, you'll see a cell, an osteocyte, and all of these white circles and lines are the cell processes running throughout the volume of the bone structure, or the canaliculi. And so we can take these 2D images, they've been put together into a 3D stack, and we can reconstruct and segment some information from this. So one of the nice things we can do is segment out the cell networks, and you can see this really widespread interconnected cell network. And we have one cell that's visible here, but there's also another cell that's outside of this data set. So we actually have the zone connecting the cell networks between two cells inside this data. And this is exciting because it starts to approach the length scale or the volume of what you could achieve if you went to a synchrotron. So this is an example on the right of nano CT or synchrotron-based CT experiments, which are fantastic and give us high resolution and large volumes. But of course, one has to travel to a synchrotron to obtain this data. So the advantage with the plasma fib is these are experiments you can do in your lab without traveling to a synchrotron. Other things that we noted from this data was that we could actually see cell organelles. This wasn't the intent of our study. We were interested more in the bone as opposed to the cells. But if your samples are maintained well enough, you can start to look at the interior of your cells as well. And of course, by having this 3D data, we can explore quantitatively other features like the size of the canaliculi, and we could perform some measurements on their size. They're around 350 nanometers in diameter, and this is really exciting because most estimates of their size are using diffraction-based techniques like X-ray tomography as opposed to direct imaging techniques. So this tool gives us a direct measurement of the size of features like canaliculi. And the second really exciting thing about using plasma Fib was we were able to do this also on tissue that we didn't remove the material, or the mineral, sorry. So in the previous slide, you'll have seen that there was very little contrast, but in this slide, you'll see really bright differences of bright white zones and dark black regions. And so in this case, we left the mineral intact, and so this is the bright zones. And anything you see in black is representing cell networks or open space, porosity. And when we look at this in a video, we can take a look at one of these planes. So we'll look at the XZ plane here, which actually aligns with the transverse plane of a femur. This video is showing us slicing through this. So we have a cell in the top left corner, and we can see these black lines that look almost like worms going through the structure. These are our canaliculi. And what we noticed for the first time was something really interesting. Everywhere else, we see these circular type features, and they're across the entire bone structure, these round rosette type features. And this is something we hadn't noticed across a huge volume in bone before. And we can see that these features are everywhere. So the mineral is really clustered into these circular features. And we had seen these before. We had seen them in a little bit of work at higher resolution. So in the red images over here, we have some features we saw in the scanning transmission electron microscope. But with the plasma Fib, we can see these features are everywhere. The TM, remember, was quite a small volume, so we didn't have a large area to look at. But with the plasma Fib, we see these features across a huge volume. The other exciting thing with the plasma Fib is we have a three-dimensional data set. So these features appeared circular in the TM, but now with the plasma Fib, we can explore what shape are they actually. And when we look at the three-dimensional shape of these features, we actually find that they're not just circular features. So they have a circular cross-section in one direction, but they're actually elongated ellipsoidal features. So you can see that circular cross-section now looking down from the top, but from the side view, we see that we actually have a mineral ellipsoid. And these ellipsoids are about 2 microns long, 1 micron in diameter, and they were ubiquitous across our bone structure. And this corresponds really well to things we've observed in TM, but didn't have the ability to view in 3 dimensions, such as rosettes that we've seen, or elliptical type motifs down other orientations. So what we really show now is that there's this elliptical structure and clustering of bone mineral that was never observed before in 3 dimensions. So we're wondering if this is a new level of mineral hierarchy. And around the same time as we published our work, our colleagues in Sweden did some scanning electron microscopy where they removed the collagen from their bone tissue, and they saw similar ellipsoidal structures. They called these marquees, and they actually saw the evolution of these structures across cranial sutures. So their hypothesis is that these are maybe a transient phase of mineral, and that the ellipsoidal structure changes with time. We were looking at a mature sample, and we saw these ellipsoids through the entire bone structure. Other colleagues at McGill University, so the group of Mark McKee and Natalie Reznikov, in the same paper or journal as us, published similar findings, but this time on wild type and hypophosphatemic mice. And what they found was that their diseased mice actually showed a deviation from this ellipsoidal structure and a really disorganized type of mineral packing. So it's quite interesting to see the observations one can make when you're employing 3-dimensional imaging techniques to see these new features of mineral clustering within bone structure. So some of our work afterwards, this was a great project to do in the pandemic. We wanted to probe all of our previous work to see, were these structures there all along and we just didn't notice them? And in fact, they were. So this is a review paper from one of my PhD students, and she took a look at a number of different biomaterials, such as bioglass, titanium implants, hydroxyapatite implants, different calcium phosphates. And what she found in all of these samples was that, again, these ellipsoidal mineral clusters were ubiquitous and they were present at bone implant interfaces as well. So they're also present in newly forming bone tissue. So our future work is really interested at looking at how these structures form and how they evolve with time, and really what is their role in supporting osseointegration at implant interfaces. So with that, I want to conclude. I showed you 3 really diverse topics today, but all on strategies to image bone implant interfaces and biomineralization events. And I want to summarize a little bit that, by using advanced 4-dimensional spectroscopy and atom probe tomography, we could have a way to probe the nanoscale and the atomic continuum at bone implant interfaces. And then with using atom probe tomography, we have a chemical approach to look at the arrangement of collagen and mineral. And lastly, this finding that these mineral ellipsoids are really ubiquitous in bone. And I think this is an exciting observation because once we understand the natural structure of bone, this should influence the way in which we design synthetic replacements. And so it would be really interesting to see if this understanding and knowledge of mineral ellipsoids impacts our ability to design better biomaterials in the future. So with that, I want to thank many people, and mostly the students who are doing all of this group. So this is the students in my research group. Some of them named here were featured in this talk for their work. Of course, all of our collaborators who we work with, and the facilities where we conduct our research, and the funding resources. And I want to send a special thanks to the organizers of this symposium for the invitation, and also the sponsors, the Canadian BiomaterialsSociety and TransMedTech. And thank you all for your time, and I look forward to taking your questions.
Well, thank you very much, Professor Grandfield, for your excellent presentation. I really enjoyed your talk. Very nice schematics and diagrams, as well as Orest also mentioned in the chat. So I just wanted to remind everyone that our next speaker is Professor Bo-Young Zhang from McMaster University, and then he will be talking about platform technologies for high throughput and high content drug screening. If you want to get most up-to-date information about these talks, make sure that you follow us on Twitter. Our handle is @TransLBME. Also, you can follow me and Human on LinkedIn and Twitter for more information about these events. So with that, I also would like to encourage participants to ask their questions using the Ask a Question box. If you don't ask, I'll ask many questions from our speaker, and then the time will be, by the time that my questions are done, then there will be no more time for you guys to ask questions. So make sure that you go first. So we have a couple of questions from the audience. We have Kibret here with us, Professor Menenquet. So thank you for the great talk. Beyond the fundamental understanding, could you please comment on how these 4D dataset could be utilized to design new materials for strong bone implant interface bonding? Very interesting question, especially addressing implant loosening over time.
Yeah, that's a great question. And I think that's the goal, right, is to improve the implant outcomes. So our initial goal was to figure out a way that we could analyze this, and we found it was quite time intensive to make this workflow, but now we have the workflow, so that's great. So now the next step in able to use it to create stronger materials is applying it to different types of interfaces. So I only showed one example, but you could imagine if you had an implant with no modification that you analyzed, as well as an implant with, let's say, a nanotube with the drugs inside of it. You could use these 4D data sets to understand If bone grows into the nanotubes, you could also use them to probe how the drug releases outside of the nanotubes, to see if the structure of the bone itself changes. So there's some evidence for different types of bone structures are more mature than others, such as new bone, which is woven and quite disorganized, or more mature bone, which has a more layered structure. So you could use these imaging techniques to look at drug release, but also to see, did the bone structure itself become a stronger type of bone, a more mature type of bone with different biomaterials underlying it. So we need to probe more.
I can't hear.
Mohsen, can you hear us? Mohsen is disconnected. So.
Okay.
Yeah, I can hear. Can you hear me?
Yep.
Can you hear me?
Yes, we can hear you.
You're back.
So I mean, okay. Sorry. I mean, there's always some technical issues with wonderful. Thanks. So the next question is from Morgan. Thank you so much for your fascinating talk. I'm particularly interested in characterizing the bone mineral content of skull bone in vivo for improved EEG, EIT modeling. I don't know if you're familiar with any techniques we can use non-invasively in patients. That was my question too, actually.
Yeah. No, that would be amazing. Unfortunately, all of the techniques I showed you today take place in a vacuum. So this is definitely something we can't use on patients non-invasively. But there are some imaging techniques where we can inject stains, but they aren't as effective at looking at the mineral. The mineral is really a challenging phase to image. So I don't have a solution for this at the moment. I hope it's something that I could perhaps think about for the future. So I think, it's still a challenge for us in the community. How do we apply these tools to clinical settings and to patients? That's a real gap right now.
So my question is, so I think Morgan asked my question, one of my questions, but my next question is about the type of materials that you can image using this. So primarily you use bone material, which is pretty hard material, biomaterial. So can you use this method to, for example, look at softer materials like gels, for example, and then take a look at the porosity of these gels?
Yeah, absolutely. So these tools can be applied to any type of material. There's some challenges as the material becomes softer. So you have to be careful about how you prepare the sample. That's the only difference. So if you wanted to look at a gel, the gel needs to be dried. So you have to think of ways to preserve its structure. So one of those options is cryo. So you can freeze the gel and look at it in cryogenic conditions using these same tools. Another option is fixing the gel and embedding it with something like a plastic resin to provide it more integrity. And then you could also apply these tools. So neither of those are perfect scenarios because you're, of course, taking something wet and making it dry or making it hard. But it is a way that we can look at different types of tissues. So one of the most imaged materials with these techniques is actually brain tissue. So this is used quite often to look at soft tissues like brain tissue as well.
Awesome. So my next question is about your in vivo probe that you talked about. I assume that this is a probe that can be used to look at these basically the bone structure in live animals. Am I right? Is it possible to use that too? Or you need to collect samples and then look at them afterwards?
Yeah, the samples are collected. So the idea is that the bone was mineralized in vivo. So most models where we're looking at high resolution are using synthetic collagen. So extracting collagen from a rat tail, placing it in a solution with simulated body fluid, and then you place that in the microscope. So we know that simulated body fluid isn't forming mineral the same way that a living being is. So our in vivo system means that the animal or the human, the patient, grew the collagen and mineral organization, and then we were able to extract a sample from that and look at it. It's not living when we're imaging it. We've taken a sample out of the patient in order to look at it.
I think we lost Mohsen again. Mohsen, are you there?
And unmute myself.
Oh, okay. So Mohsen, are you talking? Yeah, talk again from the beginning.
So you need to take biopsy samples to be able to basically look at this. Okay, that's very good. But is there any? Do you see in the future that this can be used to bones, like structures in vivo, maybe drilling a hole and then sending this as a probe inside the bone when the animal is alive, or you don't think that it's necessary at all?
Oh, interesting. I mean, I think it would be really fascinating. I think we might need to use a different type of system, a different type of signal and imaging collector, because all of these techniques rely on using a vacuum. So the electrons need a vacuum to move, otherwise they can't be focused. They move around too much in space. Same thing with the atoms that we ionize. They also require a vacuum in order to hit the detector. So right now I can't envision a way that we could create that inside of a patient. But there's other strategies for that. So X-ray based techniques are fantastic for in vivo imaging. Not at the same resolution, but there's potential that they could get there. Yeah.
Awesome. And Cupid keeps asking fantastic questions. That's a very good question. I wonder if your microscopy techniques can shed light on aging of bone. Very good question.
Oh, this is a great question, and it's actually something we're looking at in our lab as well, a little bit with my colleague Henry Schwartz, who's an emeritus professor at McMaster. And we've looked at bone that's been affected by osteoporosis, and we're actually looking at the thickness of the mineral plates, and we're seeing that there's a change in the mineral thickness as a patient ages. This is in patients with known osteoporosis, so it would be really interesting to probe this in patients without disease or with other diseases, and to see the natural change in bone. But it's quite a challenging problem because bone is always regenerating. So we never know if we're probing a site that just recently formed or if it's actually the age of the patient itself. So that's a big challenge for us.
Okay, good to know. Very good. Well, I have no more questions to ask. Hooman, do you have any questions to ask from our speaker, Professor Grandfield?
No, just want to thank,
one more question. Go ahead.
Oh, there is one more question. Professor McQuannon, thank you for your talk and for your answer to his question. Okay. So I just wanna thank you also for your very, very great talk, and I really enjoyed and learned. And I encourage all the participants. Also, I have pinned our YouTube channel under this videos that you can see, so you can subscribe to our YouTube channel so you can see the upcoming videos and the previous one that we posted there. And this talk also is recorded, and by the permission of our invited speaker, will be uploaded. I'm a little bit behind for uploading these videos, but I will do that eventually. Thank you, everybody, for your attention to this seminar series, and thanks to our speakers, and have a great day. Thank you.
Great. Thank you so much.